Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you're new here, welcome. This is a series on the chapter Biological Molecules of Chapter 1 of the STPM Semester 1 Biology series. Check out the full playlist on my channel. If you want access to flashcards on this topic and other videos, do click the link below. It is available on my Pinterest and slides on this topic are available on my website. In this video, we will be continuing nucleic acid, whereby we will dive into the differences between DNA and RNA, the DNA structure. DNA stands for deoxyribonucleic acid, and it is based on the Watson and Crick model of double helix. It's important to remember this, especially for exam. The nucleotides are of deoxyribonucleotide type, which compose of bases A, C, G, and T. A is for adenine, C for cytosine, G for guanine, and T for thymine. Now, if you want to know more about the bases and also the difference between the pentose sugar, that's how you differentiate between DNA and RNA, you can check out my previous video on this topic. So the two strands of polynucleotides are complementary to each other, meaning A is paired to T with 2 hydrogen bonds and C is paired to G with 3 hydrogen bonds. It's important to remember this, especially the amount of hydrogen bonds required to link between these two. A to T, C to G, A to T 2, C to G 3. Both strands are arranged anti-parallel to each other. In one strand, the pentose is arranged in a 5-to-3 pattern, while the other in a 3-to-5 pattern. Anti-parallel meaning they are arranged facing each other in the opposite fashion. For example, take a string. You have the head and the tail of a string, right? So a 5-to-3 pattern facing a 3-to-5 pattern, meaning the head of one string is facing the tail of the other string, and vice versa. Both strands are linked by hydrogen bonds. So here's an example. Okay, so when you combine these two strands over here, note they're arranged in 5-to-3 and 3-to-5 patterns. So you have the head facing the tail of the other one and the tail facing the head of the other one. And when they're linked together, they're linked by hydrogen bonds. Remember, A combines to T with 2 and C combines to G with 3. Okay, and then when they link together, they will spiral and you get the helix structure. RNA stands for ribonucleic acid. RNA consists of one strand of polynucleotide and is made up of A, C, G, and U. So you have the adenine, cytosine, guanine, and the uracil bases. Remember, for RNA, it's uracil. For DNA, it's thymine. So take note here, DNA you have 2 strands, RNA you have only 1 strand, meaning you only have this portion over here. So the pentose for RNA is ribose. And there are 3 types of RNA. You have the rRNA, the ribosomal RNA, mRNA, messenger RNA, TRNA, transfer RNA. First, the rRNA, ribosomal RNA. The role of rRNA is to form ribosomes. So just remember the name, ribosomal RNA, to form ribosomes. These allow the ribosome to combine with the mRNA, which is the messenger RNA, to read the codons in the mRNA and translate the codons into sequences of amino acids in the replication of protein. Basically, when they want to replicate to make new proteins, these rRNA have ribosome. Okay, they will form the ribosome, so they will combine with the other RNA to read the codons of the DNA. So they will combine together to read it first, then they will replicate, they will translate, they will replicate, they'll make more. So that's important in the formation of or replication of the protein. Then you have the mRNA, the messenger RNA. It copies the information from at least one gene in the nucleus. In the nucleus, you have the DNA, right? And it's just a very long, tangled up ball-like structure in the nucleus. So this mRNA will go over there and it will copy. It will read it first and it will copy. So it will copy the code over there. It is the longest RNA structure because you don't know how long it's gonna copy, right? So it's the longest one. It is the transcript of a gene-based sequence, and it acts as a template in which amino acids are joined into a protein of a specific kind. Basically, it's like a carbon paper. So it will go over there into the nucleus, it will read the DNA, it will copy a certain segment. So you now have the carbon copy of it, but you only have the carbon copy of the message of the code of how to make it. You don't actually have the next one. It copies the recipe, in other words. Okay, finally you have tRNA, transfer RNA. A tRNA transfer a specific amino acid to the ribosome according to the codon in the mRNA. It is complementary to its anticodon to form a protein. It is also the smallest RNA. It is complementary, meaning that after the mRNA has copied, right? Like I said, it copied the DNA. Now it has the recipe. And now this tRNA will transfer. It will be like, oh, okay, now I'm going to do the work. I have the recipe here. Now I'm actually going to do it. I'm going to follow accordingly to what mRNA gave me. So complementary meaning here, you see this part here, this is the anticodon. So remember which bases combines to which, as I mentioned earlier in DNA. Let's go back. Okay, so here A is paired to T, C is paired to G. If we come over here, so you have CAU. So C, the next one, when it forms the new one, it will form G. A will form T, U will form A. Okay, so remember in ribosome it's U, but in DNA it's T, right? So in mRNA, U is actually T, it's actually thymine. So when it forms something complementary to it, its partner, it would be A, okay? And when A, it forms its partner, it would be thymine. So just a brief summary of DNA versus RNA. In DNA, it consists of double strands, 2 strands of polynucleotide, whereas in RNA, it consists of a single strand of polynucleotide, only 1. DNA is a bigger molecule with more than 1 million bases, and for RNA, it's a smaller molecule with less than 1,000 bases. For DNA, it consists entirely of a double helix strand, 2, whereas in RNA, there's no double helix because it's only 1. The pentose sugar used for DNA is deoxyribose, and the pentose sugar used for RNA is ribose. The bases for DNA would be ACG and T, whereas for RNA, it's ACG and U. The difference here would be T and U. In DNA, there's only 1 type, whereas for RNA, you have 3 different types. You have tRNA, mRNA, and also rRNA. DNA, the function, it just carries genetic information, whereas for rRNA, it depends which RNA are you talking about. Is it tRNA, is it mRNA, or is it rRNA? And each of them have their own important role in terms of protein synthesis. DNA is found mainly in the nucleus, whereas RNA is found mainly in the cytoplasm where the protein synthesis actually takes place. If you've reached this point, thank you very much. Give this video a like if you found it helpful, as it does help in reaching out to others as well. Do support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video to those who might find this helpful. See you in the next one.